Yeah. So the thing is that, Siddhant, we will begin with our lecture. I got your homework, dear. Okay, good to know that you have completed the homework on time. Definitely now I'll be checking up with your homework. Wait a second. How you found with the questions, whether it was easy or whether it was hard, Siddhant, for a homework? Easy. It was easy, dear? Okay, okay. All right. So the thing is, Siddhant, yeah. Yeah, Siddhant, very good, beta. All right. Okay, Siddhant, one more thing I would like to tell you, dear. That is, you draw the diagram that is after writing the answers, right? Okay. Yes, only the thing is, Siddhant, you have to do one thing. You have to draw the diagram before writing the answer. Yeah. Okay. And always remember, Siddhant, the board rule is that you have, I saw one thing, the labeling has been done little bit randomly, right? So the thing is that the labeling which has done randomly, you shouldn't do it randomly. Okay. The labeling should be always on a one side. You decide it whether you want a left hand side or you want a right hand side, which side you are feasible with, right? According to the board rule, they are given as in left hand side for labeling the diagrams. But now the thing is that some of the students, they are like, they are feasible with right hand side. Some of the students, they are feasible with left hand side. So moderator says to the examiner, see towards the students that they are labeling at a one side. That's it. Okay, so whichever, if you feel that the cell is very far away or something, please take a scale, draw a line in between of the diagram, it's fine, but try to have a labelings on one side. Next thing is that there, for example, like question number 5, if we are telling, or question number 3, I'm telling you, they draw a neat and label diagram of male gametophyte. Okay, so what you are going to do, dear, when you are drawing about the male gametophyte, now you have mentioned the question, that is question number 3. Some of the students have habit of writing, draw the label, neat and label diagram of male gametophyte. ये आपने पेन से लिखा क्योंकि वो क्वेश्चन है आपके पेपर में. आपने डायग्राम बनाया, डायग्राम बना के बाद उसको सारे एक साइड में लेबलिंग कर किया, और उसके बाद भी नीचे पेंसिल से ही उस डायग्राम का नाम वापस लिखना. ओके, क्यों? क्योंकि ये इम्प्रेशन बहुत अच्छा देता है. कभी कभी अगर कोई कोई एग्जामिनर्स, इफ दे आर लाइक, इफ दे आर नो मोर इन अ मूड टू गिव द मार्क्स, द थिंग इज व्हाट, they cut down the marks of the students. Why? Because they have written up with the help of a pen, but they have not mentioned with the help of a pencil. Okay, so it is not a presentative diagram into a board exam, isn't it? According to the answers and everything, it's very good, and the diagram has been also drawn very well of yours, very precise answers and diagram. All right, I'm happy to see that. Only the thing is that is Siddhant, you have to draw the diagram first and then write down the answer. Okay, bacha, and try to draw all the labelings on one side. Okay, beautifully done the diagram of in birding also. I like that the way you have done it. All right, and even the pollen crane. Okay, anther wall and the dispersion, that is undehized anther, which is a dehized anther. Beautifully drawn, beta. All right. Okay, chalo. Now let's begin with our lecture, dear. I'll share a screen. Yes, with. Yeah. Yeah, please, please, please. Okay, Siddhant. Siddhant, can you see my slide? Siddhant, is the slides have been seen, dear? Yes, ma'am. Okay, all right. Okay. So the thing is, we have already studied with reproduction, asexual reproduction, mode of asexual reproduction, right? And in the last lecture, guys, we have studied with the what sexual reproduction in plants, right? So the sexual reproduction in plants, we saw which are the whorls which are participating into a sexual reproduction, and which are the essential whorls and non-essential whorls. Then we studied about a typical presentation of the flower, which is this one, right? And then even we have studied the next diagram, which are representing the stamen and carpel. Okay. Then we went toward a structure of anther. And this structure of anther, we have studied the androecium, stamen, diathecious. Even we have studied about this tetrasporangian, monothecious, right? We studied about the 4 layers of anthers, that is epidermis, isn't it? Which is the next Siddhant after epidermis?
Endothelium.
Yes, then?
Middle layers and then tectum.
Yes, very good. And we have saw that which how this layer are functioning and doing their work, isn't it? Then we saw towards a structure of anther, right? That is a stamen is including anther and filament. Then even we saw the undehisced anther and the dehisced anther, isn't it? Whenever the anther get mature, how the pollen grains get mature, and even they are ready to dispersed. Then we went towards a diagram that we can label it, and in this way it can be asked into the exam, right? It was a practicing diagram we have seen. Then we begin with a microspore, which we called it as in pollen grain. And in this, we studied about a microsporogenesis just in gist. And even we studied about a megasporogenesis and a structure of a microspore we studied, right? Then we started about what? Then we started about a structure of the pollen grain, that is a male reproductive body. Okay. And then we saw how many coats are present, isn't it? Then we saw that the chemical is produced by tapetum, but it is present on the hexine. Even we saw that in how many monocot produces the germ pore. This germ pore is further going to get developed into a germ tube, which we also called it as a pollen tube. Therefore, this is a structure of an pollen grain, right? In this pollen grain, majorly we studied the cell of vegetative nucleus, then generative nucleus, and even we have studied the generative cell. Then we saw the structures, that how is it formed, how the germ pore are found into the compound microscope. That is how many germ pores are present in monocot and dicot. And even this slide, it has been made by me, that is, and even by the students, you all like students, that how these pollen grains are emerging out the pollen tube, isn't it? Then we went towards what? We haven't went, but today we are going to study about development of the male gametophyte. Isn't it, Siddhant? Yeah.
Yes.
Okay. So dear, this male gametophyte which we are going to study, the male gametophyte which we are going to study, dear, right? Siddhant, you write it down, this one. This is visible to you?
Yes, ma'am.
Yeah, write it down. All right, Siddhant, may I start, dear?
Ma'am, ma'am, we are while you are teaching, I can write it a little bit.
Okay, it's okay, it's okay. You can write it down. All right. Okay. So the thing is that which I'm going to talk today, that is a development of male gametophyte. That how this male gametophyte have been developed, right? The male gametophyte into our what? Into our, that is into our a plant flowering structure. We are looking towards a what? We are looking towards a pollen grain. Therefore, this male gametophyte is nothing but that is a microspore. And this microspore, Bacha, we are also calling it as an initial cell of the pollen grain. Initial cell means what? The starting cell which is formed into the anther, and hence furtherly it is going to help for the fertilization purpose. So we called it as in what? We called it as a male gametophyte. This male gametophyte is endosporic, right? Definitely we have seen into an anther where we have saw into a pollen grain, okay, that we have saw that it is a heterospory. Okay, when we started about the sexual reproduction, we have studied it as an heterospory. Heterospory, why? Because we have a two different spores formation, that is male spore and female spore, right? That's why it has been said what? The male gametophyte which is present, it is what? Endospory, because it is present inside the plant cell wall. If it would be outside, we would have said that is it is an hexospory, right? Therefore, dear, what we have saw that into this male gametophyte Siddhant, it involves a two mitotic division. How many stages? Two mitotic division stages, right? Therefore, these stages have been completed into a two stages, that is before pollination in pollen sac. Whichever this male gametophyte activity will be carried out, it has been divided into how many parts? It has been divided into two parts. The firstly, it is what? Before pollination in pollen sac. So before pollination, whenever the pollination, what is the meaning of an pollination? Pollination means the depositing of pollen grain on stigma. बराबर? तो ये जो पोलन ग्रेन जब स्टिग्मा पे डिपॉजिट होगा, तो होने से पहले ये पोलन सैक कैसे होता है, क्या प्रोसेस होगा, ये सबसे पहला स्टेज है. एंड व्हाट इज द सेकंड ईयर? सेकंड इज आफ्टर पॉलिनेशन ऑन द स्टिग्मा. स्टिग्मा पे पॉलिनेशन होने के बाद आगे की प्रोसेस कैसे चलेगी ये मेल गैमेटोफाइट की, वो हम देखेंगे. ओके, नाउ let us move further and see that is before pollination in pollen sac. Now, before pollination in pollen sac, that is the male gametophyte is nothing but it is our pollen grain, right? And therefore, this male gametophyte starts, means what? It is starting under the pollen grain into the pollen sac. And this pollen sac is present within the anther. Am I right, Siddhant? Anther wall के अंदर हमारा पोलन सैक के अंदर जो है हमारे पोलन ग्रेन्स, जो कि एक्ट करेंगे हमारे मेल गैमेटोफाइट के तौर तरीकों से. बराबर? अभी this pollen grains, okay, they are going to go under a mitotic division. Cell division is divided into how many parts? Two. That is mitotic and meiotic. बराबर? That is mitosis and meiosis. माइटोसिस अगर आप देखते हो तो उसमें आपको 2 डॉटर सेल्स मिलता है. मियोसिस आप देखते हो तो उसमें 4 डॉटर सेल्स मिलता है, right? So in same way, the thing is that in mitosis, that is what we are going to get a two male gametes, right? That is a two daughter cells. So we will see that how these two male cells and the two daughter cells are going to get towards us, right? जब pollen grains divide होता है mitosis से, right? When pollen grains start dividing by mitosis, there is a production of two unequal cells. ऑल राइट, ये पोलन ग्रेन क्या करेगा? डिवाइड करेगा माइटोसिस से, माइटोटिकली डिवाइड करेगा और कितना सेल्स बनाएगा? टू अनइक्वल. द सेल साइज इज नॉट इन इक्वली साइज, दे आर इन अ अनइक्वल साइज. Now what we will see? We will see that the cells which are formed, two unequal cells, they are in a present of a larger cell and they are present in a smaller cell. ओके, जो दो सेल्स फॉर्म हुए ना सिद्धांत, एक बड़ा सेल बना और एक छोटा सेल बना. ओके, द लार्जर सेल इज कॉल्ड एज इन हियर वेजिटेटिव सेल. इट इज आल्सो कॉल्ड एज एन ट्यूब सेल. एंड हेंस यही जो वेजिटेटिव सेल बनाएगा, ये क्या बनाएगा? ये न्यूक्लियस भी बनाएगा. और जब ये वेजिटेटिव सेल न्यूक्लियस बनाएगा, उसे हम बुलाते हैं वेजिटेटिव न्यूक्लियस. ओके, एंड टू सेल्स बना. मैंने सिर्फ आपको लार्जर सेल के बारे में बताया. स्मॉलर सेल इज गोइंग टू फॉर्म व्हाट? इट इज गोइंग टू फॉर्म अ जनरेटिव सेल. And this generative cell is also going to form a nucleus, Siddhant. So we are going to call it as a generative nucleus. When these two cells are formed, that is generative cell and sorry, that is what? That is a vegetative nucleus and generative nucleus. We called it as an what? We called it as an two cell stage pollen grain. Okay. Understood, Siddhant? Mitotically divide हुआ pollen grain, माइटोटिकली डिवाइड होने के बाद 2 सेल्स फॉर्म हुए। 2 सेल्स फॉर्म हो जाने के बाद क्या हुआ, एक सबसे पहले बना जनरेटिव सेल। जनरेटिव सेल ही न्यूक्लियस बनाएगा, तो वो जनरेटिव न्यूक्लियस बनेगा। वेजिटेटिव सेल जब उसका खुद का न्यूक्लियस बनाएगा तो हम उसे बोलेंगे वेजिटेटिव न्यूक्लियस। जब ये दो टाइप्स के सेल्स बनते हैं, जनरेटिव एंड वेजिटेटिव, वी कॉल्ड इट एज एन 2 सेल स्टेज पोलन स्टेज। अंडरस्टूड डियर? समझ में आया आपको? इसीलिए आपको ये भी समझ में आया इंटाइन के अंदर जो साइटोप्लाज्म का प्रेजेंस है, बराबर, मतलब जो सारे सेल्स प्रेजेंट है हमारे पोलन ग्रेन में, राइट, द हेक्साइन इज अ आउटर कोर and intine is a inner core, and inside the intine we have a cytoplasm, means what, it is a cell house, बराबर। तो उस सेल हाउस में हमारे पास वेजिटेटिव न्यूक्लियस है, जनरेटिव न्यूक्लियस है एंड जनरेटिव सेल प्रेजेंट है, बराबर। So in this way we have a vegetative nucleus and we have a generative nucleus, which are our 2 cell stage pollen cells. All right, Siddhant, till here have you understood? Siddhant, have you understood
Yes,
here?
ma'am.
Okay, chalo. Now let us begin further.
Ma'am, can I write that?
Yeah,
Write that?
yeah, for sure, dear. You can write it down. Please write. Siddhant, once it is done, please let me know, okay, dear?
Done, ma'am.
Done, bacha? Okay. Now, when this 2 cell stage, when it is formed, when pollen grains become a 2 cell stage, what happens? The anther wall get ruptures, because the major thing is that what? The major thing is that the formation of pollen grain, and the pollen grain should get mature, right? So over here the pollen grain has got mature, Siddhant, okay, because all the pollen grains have reached towards a 2 cell stage, 2 cell stage, right? सारे pollen grains में, anther walls में सारे pollen grains क्या हो चुके हैं? 2 cells बन चुके हैं, right, via a mitotic division. तो अब anther wall क्या हो जाएगा? Anther wall has got mature and it is going to get ruptured and it is going to get broken down, right? And who is going to break that anther wall when it is going to get ruptured here?
Endothecia.
Endothecium, absolutely right, beta. And therefore, what? The pollen grains are free to disperse. Now, this was before the pollination, what the activity has formed. Okay. Now, the further thing is how the pollen grain has been developed into the male gametophyte, into a proper male, that is, into a proper androecium, right? So, the second stage is that after pollination on the stigma, तो जब स्टिग्मा पे पोलन ग्रेन प्रेजेंट होता है तो आगे जाके उनकी पोलन ट्यूब कैसे बनेगी और वो आगे जाके कैसे मैच्योर होंगे, राइट? और उसी पोलन ट्यूब के मदद से वो ओवरी के तक कैसे कैसे पहुंचेंगे, right? And inside the ovary there is a presence of an ovule, and from that ovule how are they going to reach towards the embryo sac? Understood, Siddhant? So in this way, how the activity is going to carry out, we are going to see into the after pollination on the stigma. These pollen grains are what they are pollinated, means they are dispersed with the help of an what? With the help of an different pollinating agents. Now, who are our pollinating agents? Our pollinating agents is wind. Our pollinating agent is insect, even via water, even via birds, right? These all are the pollinating agents, dear, isn't it? But once the pollen grain is mature, right, they have been released from the anther wall, all right, now they have deposited on the stigma. And after depositing on the stigma, what these pollen grains do? These pollen grains start absorbing a nutritional substance, that is a sugary substance. ओके और ये शुगरी सब्सटेंस कहाँ पे प्रेजेंट है? ये शुगरी सब्सटेंस प्रेजेंट है स्टिग्मा पे. Okay, Siddhant. So whichever the pollen grain is of its own, मतलब जो कोई भी plants का खुद का pollen grain stigma पे present है, वो क्या करने वाला है? It is going to provide what? It is going to provide a nourishment to that pollen grains. And what it is going to do after absorbing that nourishment, that is sugary substances from stigma, they are going to get swell. And after swelling, what it is going to happen? The intine is going to push out, right? And once the intine is pushed out, definitely the hexine is going to push out. And the hexine is pushed out, there is a present of a minute pore formation, and that pore we called it as a germ pore. Understood? And that germ pore is further going to form what? The pollen tube that we called it as a germ tube. Understood, dear? समझ में आया सिद्धांत यहाँ तक कैसे हमारा germ pore बाहर आया? Okay. Hence, now the generative cell which is present, okay, towards your tube nucleus. Okay. Now what this generative cell is going to happen? This generative cell with its nucleus, it is going to travel, means it is going to migrate towards a pollen tube. बराबर हमारा pollen tube बन चुका है अभी. तो वो पोलन ट्यूब अब इमर्ज पूरा क्या होगा? बड़ा हो जाएगा, स्टाइल से पास ऑन होगा. एंड आफ्टर पासिंग टुवर्ड्स दिस स्टाइल, इट इज गोइंग टू रीच टुवर्ड्स द ओवरी. एंड ओवरी इट इज गोइंग टू टेक इन टू दैट टुवर्ड्स द ओव्यूल, राइट? तो वहां तक पहुंचने के बाद वो क्या होगा? पोलन ट्यूब फॉर्म होने के बाद जनरेटिव सेल क्या बोलेगा न्यूक्लियस को? चलो तुम भी चलो, अब हम चलते हैं, राइट? एंड दे ऑल आर गोइंग टू ट्रैवल, दे ऑल आर माइग्रेटिंग टुवर्ड्स द पोलन ट्यूब. Right. Now this pollen tube is growing in a downward direction because stigma is apposite, right? Pollen grains उस पे deposited है. Nutrition खुद का pollen grain होने की वजह से absorb हो चुका है. Pollen tube properly immersed हो चुका है. अब ये properly immersed होने के बाद ये downward direction में जाकर it is reaching towards whom? It is reaching towards the ovule. Okay. And once it is reaching towards the ovule, साथ में हमारा generative cell जो है, right? This generative cell is going to start dividing itself via mitotic division, right? And when this generative cell is going to divide itself via mitotic division, it is going to form a two haploid cells. And these two haploid cells are nothing but our two male gametes. Okay. And these two male gametes are in a non-motile condition. They don't have any kind of a motile condition. They don't have any kind of a movement. जैसे जैसे pollen tube बढ़ेगा वैसे वैसे ये male gametes उसके साथ travel करते जाएंगे. Okay. Now further what it is going to happen, further development is going to carried out, and nucleus which is present, the nucleus which it has been traveled with generative cell, जो generative cell के साथ nucleus travel किया, वो nucleus धीरे धीरे degenerate हो जाएगा, गायब होने लगेगा, and what it is, they will that nucleus will tell to the male gamete, you remain as it is into the pollen tube, because it is representing it is consisting of a male gamete into a pollen tube. So the pollen tube is filled with the two male gamete, and therefore it is representing that yes, this pollen grain is what? It is a androecium. It is having a what? It is having a quality like a male, and hence it is called as, and in this way the formation of male gamete is taking place. Understood, Siddhant? यहाँ तक आपको समझ में आया? इसलिए मैंने जब स्टार्टिंग में जब मैंने आपको पोलन ग्रेन का स्ट्रक्चर दिखाया था, तभी मैंने आपसे एक चीज कही थी, that is what I said you, generative nucleus and generative cells both are interlinked with each other, right? Now understood why are they interlinked with each other? Because करने के लिए तो vegetative nucleus भी function कर सकता था, but vegetative nucleus, what he said, it said that I'll remain as it is, जो करना है तुम दो दो हो, तुम कर लो जो है वो, बराबर? इसीलिए the generative cell got functional, and the generative got functional, and generative cell said we are two, right? So what we will do, we will go under a mitosis condition and we will form a two haploid cells. Understood? And therefore the two haploid cell which is present, it has been what? It has been formed, and therefore they are reaching towards whom? They are reaching towards a pollen tube. Understood, Siddhant? समझ में आया कैसे दो मेल गैमेट्स फॉर्म हुए? Now further we will move. These two male gametes are always in a functional, and they are even going to play a important role into a double fertilization and in female gametophyte. Now you can see this diagram, that is development of male gametophyte. Now this is what? This is a single microspore. And the single microspore which is present, it consists of hexine, it consists of entine, and it consists of the normal nucleus, as all the cells consist of. Now in a second stage, what we have saw that definitely what? The pollen grain have been deposited, the pollen grain have been deposited on the stigma. It has absorbed all the nourishment, right? So after absorbing the nourishment, after absorbing these sugary substances, the intine has been pulled out. And due to the intine has been pulled out, the hexine has been pulled out. Barabar? And in that way, on the hexine, there is a presence of a germ pore. बराबर? And that germ pore is forming a pollen tube. लेकिन उसके पहले जो बिफोर पॉलिनेशन में हमने देखा था, हमारे पास माइटोसिस के थ्रू हमारे पास दो सेल्स बने, and though two cells was larger than smaller, that is generative cell and vegetative cell. So this stage has been happened, what? This stage has been happened during the before the pollination. After the pollination, when it has been reached, what we have saw, that the pollen grain has absorbed sugary substances, pollen tube has been immersed out, and therefore what has happened, this generative cell has divided into the two cells, right? Via which? Via a haploid condition, via a mitotic condition, right? So therefore what? These two cells which are divided, therefore there is a presence of a tube nucleus. Now what this tube nucleus is doing? As this pollen tube is growing towards downward condition, it is what? This tube nucleus, it is showing a pathway to the male gamete. And once the male gamete has been reached properly to the pollen tube, this tube nucleus is going to get degenerated. And therefore the male gamete is going to remain as it is. And hence this process is called as in development of the male gamete, and it is present into our pollen grain. Pollen grain is present into a pollen sac and into a anther. Hence the whole structure we called it as in stamen with anther and filament. All right, Siddhant.
Yes,
Have
ma'am.
you understood till here, dear?
Yes, ma'am.
Any kind of inquiries till here, dear? Understood? Okay. Now let us move forward.
Ma'am, can I write that slide?
Yeah, please, you can write it down here.
Done, ma'am.
Done, dear? Okay. Now, this was a development of the male gamete, the major male gamete, which is important into a sexual reproduction, right? Now, for a sexual reproduction, only one sex cell is not enough. It is also necessary to have a female gametophyte. Now, let us begin with the structure of an anatropous ovule, right? Now, we everybody know that into our flowering plants there is a presence of an ovary. Inside the ovary there is a presence of an ovules, and this ovules is into a functional stage. Therefore, it is what? It is a embryo sac, right? Now let us begin with the structure of anatropous ovule. Now, this anatropous ovule, Siddhant, which it is present, you can see it is in a downward position, right? It is slanting in a downward direction, and there is an entry, there is an opening which is present. It is from downwards. So whenever there is whenever there is an opening of the ovule in a downward direction, we called it as an anatropous ovule. Means whenever the pollen tube is going to enter from a micropyle region, that is from a downward direction, we called it as an what? We called it as an anatropous ovule. Even into our nature, we have a orthotropous ovule. Whenever this micropyle is at an upper direction, okay, at an upper stage, the micropyle, which is the entry of the pollen tube, which it is going to get from downwards, if it is going to get from the upper direction, hence we called it as an anatropous ovule. We have a next types of an ovules, that is what? We have a amphitrophus, we have a hemitropus, we have a campylotropus, and even we have a cernitotropus, right? This all are the types of our ovule, but this all the thing we will be studying under a NEET. Now the board, according to the board point of view, we will be studying about the anatropous ovule. And hence, for your extra information, I will be studying about the hemitropous ovule also. Now the thing is, Siddhant, that is, in this anatropous ovule, let me introduce the individual cell and structures. The first is what? It is an helium, right? Then we called it as an funicle. Funicle is what? Funicle, it is a stalk. Stalk means each and every, like for a flower. We have a pedicel. Okay, in a common language, people called it as in flower stick. But can we call it? We cannot call, because we are a proper specialist of the botanist and zoologist, right? So we will tell it as in stalk, or we will call it as in pedicel of the flower. In the same way, this ovule also has their own stalk, okay, which we called it as in what? In the ovule language, into a flowering language, we called it as in funicle. From whenever, okay, from wherever there is an opening of the ovule, from that opening, the the pollen tube is going to enter, we called it as in micropyle. And hence it is also called it as in micropylar end, क्योंकि वो ऐसा एंड है जो कि स्मॉल ओपनिंग है और वहां से हमारा पोलन ट्यूब एंटर होने वाला है और ये न्यूसैलस मतलब हमारे एम्ब्रियो सैक तक पहुंचने वाला है. ऑल राइट, इसलिए हम उसे कहते हैं माइक्रोपाइल. जहां कहीं पे भी ओव्यूल में माइक्रोपाइलर रीजन है, माइक्रोपाइल एंड है, वहां से हमारी पोलन ट्यूब की एंट्री होगी पोलन हमारे ओव्यूल तक पहुंचने के लिए. ओके, then we have a micropyle. It also consists of an micropyle pole. All right. Then we have a two types of an integument. First one is inner tegument, which is present inside, and then we have a outer integument, which is present outside. Okay. This integument, it is giving protection to our embryo sac. ऑल राइट, ये क्या कर रहा है? ये एम्ब्रियो सैक जो है, फीमेल गैमेटोफाइट है, जो कि हम थोड़ी देर बाद देखेंगे, उनको वो क्या कर रहा है? प्रोटेक्शन दे रहा है. ऑल राइट, देन वी हैव अ न्यूसैलस. When the embryo sac is in a younger stage, we called it as a nucellus. All right, and this nucellus, it is in a quite functional stage, because into this embryo sac we have a different types of a cell, which we are going to study soon into a female gametophyte, that is antipodal cell, egg cell, synergids, polar nuclei. So this nuclei helps for all those cells which are belonging to the embryo sac to become into a functional stage. तो एक चीज याद रखना है सिद्धांत, ये जो न्यूसेलस है यही एम्ब्रियो सैक है, लेकिन जब हमारा एम्ब्रियो सैक यंग स्टेज में होता है तब हम उसे न्यूसेलस कहते हैं। और जब ये न्यूसेलस मैच्योर हो जाता है, जो मैंने सेल्स के अभी नाम बोले, सिनर्जिस, एंडोपोडल सेल्स, सब सेल्स के जो मैंने नाम बोले, ये सारे सेल्स जब हमारे एम्ब्रियो सैक में आ जाते हैं तो हम उसे बुलाते हैं अ प्रॉपर मैच्योर एम्ब्रियो सैक। All right, and then we have a chalazal pole, which is what it is a base of our ovule. Okay, हर कोई चीज के पास base होता है, right? Always the base should be strong. So wherever there is an opening of this ovule, we called it as a micropyle end. We called it as a micropyle. But wherever there is a base, we called it as a chalazal pole, that is chalaza. Okay, and wherever there is an ovule stalk, जहाँ कहीं पे ovules की डंडी है, हम उसे बुलाते हैं funicle. All right, here helium is related to the ovule placenta. All right, it is a protective covering, and therefore it is what we called it as an helium, which is interlinked with our placenta of the ovule. Understood, Siddhant?
Yes,
Okay,
ma'am.
now let us move further. Now, this ovule, this anatropous ovule, we are going to study, right? The first thing is that this is a female reproductive floral ball, and therefore you know that we called it as an gynoecium. Now, this gynoecium we include as an carpel, and this carpel consists of an ovary, style, and stigma. Ovary consists of an ovules which are not in a single stage, but it is present into a many more in it. There are many more ovules are present into the ovary. All right, and this ovary is what the ovule which we called it as an, it is one of the female gametophyte. Like into a anther, we have a pollen grain, we called it as a male gametophyte. It develops male gametophyte. In the same way, the female gamete develops what? It develops a ovule. Into all the flowering, the seed bearing, and the fruit bearing plants is called as an angiosperm. सारे एंजियोस्पर्म में अगर आप देखोगे सिद्धांत तो एनाट्रोपस ओव्यूल ही आपको मिलेगा. All right. Now, this anatropous ovule, which we are going to study, it is divided into two major parts. First one, it is called it as an funicle, and second one, we called it as an body of ovule. Now, let us begin with the funicle. All right. Now, this funicle, which we have said, we also called it as in stalk, which I said you while introducing the diagram itself. तो ये जो हमारा स्टॉक है, ये क्या है? ये ओव्यूल की डंडी है, बराबर. अगर कॉमन लैंग्वेज में कहो तो ओव्यूल की डंडी, लेकिन इट इज व्हाट? इट इज अ स्टॉक, right, which we called it as a funicle. This funicle in some of the plants it is variable in length. कोई कोई फ्यूनिकल क्या होता है? शॉर्ट होता है. कोई कोई फ्यूनिकल होता है बड़ा होता है लेंथ में. सो इट डिपेंड्स, right? So hence this funicle is what? It is variable in length. Now in some of the ovule it consists of a funicle, some of the ovule which doesn't consist of a funicle. So the ovule which consists of a funicle, we called it as a funiculate ovule. And the funicle which is, and the ovule which is without funicle, we called it as a sessile ovule. Understood dear till here? यहाँ तक समझ में आया सिद्धांत? कुछ डाउट है यहाँ तक सिद्धांत आपको? समझा? You want to write it down? You written?
Yes, ma'am. Yes, ma'am.
Okay.
I want to write it.
Yeah, write it.
Done, ma'am.
Done, beta? Okay.
Yes.
All right. Now the next thing is what? The thing is that what is a see? Now we have studied over here about a funicle, right? So majorly, what is a base? What is a function of the funicle? The function of the funicle is it always remain attached to the body part of the ovule, majorly to the placenta. ओके ये फ्यूनिकल क्या करेगा हमेशा ओव्यूल के बॉडी पार्ट से कनेक्टेड रखेगा और प्लस प्लासेंटा से हमेशा कनेक्टेड फॉर्म में रहेगा. Now the second is what? Second is body of ovule. Now in this body of ovule, we are going to study about integument, we are going to study about micropyle, we are going to study about chalaza, and we are going to study about the nucellus and embryo sac. Okay, now let us begin. Now, the body of ovule has been majorly divided into many more following parts. Firstly, I'll start with the integument. This integument, if you will see Siddhant, it is protecting what? It is externally, it is internally. But it is protecting whom? It is protecting to our nucellus. All right. So therefore, externally nucellus is covered and protected by one or two covering of the integument. If you will see this diagram, dear, if you will see this diagram, this internal part, which is a major seems to be like an oval part, we called it as in what? We called it as an embryo sac, right? This embryo sac, which it is present, it is having a outer covering and inner covering, right? This outer covering, which it is present, we called it as in what? This whole of this outer covering, we are calling it as an outer integument. And if you'll see this inner integument, which it is internally present, which it is halfly surrounded, so we called it as an inner integument. So both this outer and inner integument, they are giving the protective covering to whom? They are giving this protective covering to the nucellus, that we called it as an embryo sac itself. All right, dear. Now the next thing is, whenever if there is a one single integument present, we called it as an unitegmic ovule. But whenever if the ovule is consisting of an two integument, we called it as an butegmic ovule. Understood, Siddhant earlier? So how many anatropous ovule have the integuments, dear?
Why not?
Yeah, but basically now I shown you the diagram. उसमें आपको कितना integuments देखा बेटा?
Two.
Two. तो unitegmic हुआ या butegmic हुआ?
Butegmic.
Very good. Now, what is a function of this integument? The function of this integument is providing protection to the nucellus. Okay. Dear, there is a spelling mistake over here, so I'll correct it. Wait a second, okay? Yeah. So this was about a nucellus. All right. Now, if you will see towards a further, that is a micropyle. Into a micropyle, when you will see, that is what? It is a one of the opening, that is a lower opening from where the pollen tube is going to enter, right? And therefore, what we are calling it as an, it is an micropylar region. We also called it as an micropyle. Now, integument is not surrounded fully with a nucleus. When I was showing you the structure of integument, that time itself I show you that these two integuments, if you will see, these two of the integuments which are present, even this layer of the integument which it is present inside, okay, whole of this what they are surrounding the embryo sac, that is our nucellus, but it is halfly surrounded. It is not wholly surrounded, right? See, if you can see this outer integument, this is wholly surrounded, isn't it? But when you will see this certain kind of an integuments, it is what it is doing, it is just halfly surrounded, not fully surrounded here. Okay, so therefore what they want to say, they want to say that this micropyle region, okay, it is what? It is not fully surrounding what? It is not fully giving the protection, but layer which is formed, it is what? It is not fully surrounding the nucellus. Because why is it so? The reason behind that, it is for the micropyle and even for the integument, that is what? It is having a small opening at the base of the ovule. right? And when there is a presence of a small opening at a base of the ovule, we called it as an micropyle. And this is a reason that is why the micropyle and why these integuments are having a half covering. Though the integument outer covering is full, but inner covering, if you will see, Siddhant, it is halfly covered and it is giving halfly protection. Definitely is doing a function, but how the layer is formed, it is halfly. Why? Because it is giving a small opening to the micropylar region. What is the function of the micropyle? The function of the micropyle, that is what giving the passage to the pollen tube during the fertilization so that it can reach towards a ovary easily. Then we have a chalaza. Now this chalaza, what I already said you during the introduction of the diagram, that is this nucellus. It is a strong base of the what? It is a strong base. It is providing a base towards whom? It is providing a base towards our ovule. So the function of the chalaza is providing a base to the ovule and even a base to the nucellus. Okay, and hence the cell which provides a base to the nucellus and to the ovule, we called it as in chalaza. Wherever the chalaza is present, giving a base, we called it as in what? We called it as in chalazal end or chalazal pole. Understood, Siddhant here? Now the next we have, that is a nucellus. Somewhere around on in a some in some of the parts I have written the nucleus. Dear, it is a mistake which have been done by me. So please you should write it as a nucellus. Okay.
Before that, can I write the previous slide?
Yeah, you can write it down, please. Is it visible, dear?
Yes, ma'am.
Okay.
Done, ma'am.
Okay, dear. Now the next thing is nucellus. Nucellus, it is a major part of our ovule because during the young, when the embryo sac is young, we called it as a nucellus. And this nucellus, when it is into a mature stage, when it attend all of the cells, we called it as an embryo sac. That's why it has been called as it is a major part of the ovule. Now this nucellus, it is majorly made up of parenchymatous tissue, diploid parenchymatous tissue. Right. Now, therefore, dear, hence we saw into a diagram, this nucellus, instead of telling nucellus, I'll tell our ovule, right? Our ovule consists of in two ends, right? And those two ends are, one is at the upper side, which we called it as in chalaza. Hence, it is also called as in chalazal end. And end we have a opening, we called it as in micropyle, which we also called it as in micropylar end. Now, the reason behind why it has been putted under the nucellus, the reason behind that is the nucellus, it is a embryo sac, it is a major female gametophyte, right? So that's why these two ends are playing a dominant role in this nucellus. Now, what is the function of this nucellus? This function of this nucellus is it's developed into a embryo sac, and hence we called it this nucellus as an embryo sac or it is also called it as in female gametophyte. Understood, Siddhant, till here? Any kind of in doubt, dear? Understood. Before we studied with what? Before we studied with your writing part is over, dear?
Not of this slide.
Yeah, write it down so that I'll revise it: male gametophyte, female gametophyte, and I'll start with the embryo sac. Okay, so that it will be interlinked, dear. You can write it down full about this whole slide and you can tell me, ma'am, it's over, right? So
Okay,
yeah, it will be easier me for explaining of embryo sac, because first I'll revise male gametophyte with you.
done, ma'am.
Done, dear? Okay. Now, dear, the thing is that let us revise before. I'll ask you a few questions, right? We studied about the male gamete. Okay, Siddhant? So, the male gamete have been divided into how many stages, dear?
Two.
Tell me, name them.
The before the pollination and after the pollination of the stem.
Yes, dear. Okay. Now, you tell me that into a pollen grain, how many cells are produced?
Ma'am, how many cells are produced? Three
Yes.
cells.
Okay, name them.
Generative cell, generative nucleus, and vegetative cell.
Very good. Now, into before pollination in pollen sac, dear, there are the presence of how many unequal cells?
Two.
Two. And they are dividing
Mitotically.
mitotically. Okay, fine. Which is a larger cell
The vegetative cell.
and smaller cell?
Generative cell.
Very good. After pollination, okay, on the stigma, all right, there is a presence of what? There is a presence of a germ pore, all right. Now, when this germ pore, which is formed, okay, the generative cell undergoes a mitotic division. Which of these two cells are formed? Can you name them, those two cells?
Ma'am, the tubes, the polar tube cell and the two male gametes.
Yes, absolutely right. Are the male gametes are motile in condition?
No, ma'am. They are
Then,
non-motile. Ma'am,
okay,
the tube cell finds the pathway and through that pathway the male gametes reach the ovary.
very good. Now, tell me this anatropous ovule. What is the meaning of an anatropous ovule?
Ma'am, here the micropilar end is upside down. It is below, not above.
Okay. Okay. Therefore, now you tell me, dear, that the anatropous ovule which we have studied, it is majorly divided into how many parts?
Ma'am means integuments, etc.
Yeah.
Integuments, nucellus,
Hmm.
then the nucellus, micropilar end, and chalazion end.
Yes. All these ends, all these parts are included under which of the parts? See here. What was my question, dear? Anatropous ovule, the ovule which we have studied, majorly
Funicle and
divided.
body of.
Yes. And you said me the part of the body of the ovule. Okay. Now tell me, Siddhant, that is when we studied about funicle, if the ovule is consisting of a funicle, it is called as an,
Funiculate ovule.
and if the funicle is not,
Sessile ovule.
sessile. Okay. If the integument is present in one in anatropous ovule, you will call it as an,
Unitechnic ovule.
and when it is present too,
Eutechnic ovule.
and the anatropous ovule is common in which of the plants?
Angiospermic plant.
Yes, angiospermic plants. Very good. Dear. Okay. Can you tell me the function of the nucellus?
Ma'am, the nucellus is the young form of the embryo sac.
Hmm.
It further develops to form the embryo sac. Means it is the ovary of the it is the ovule of the embryo.
Okay. All right. Okay. Now, the majorly function of the integument is to give the,
Protection.
yes, to the nucellus. All right. Now, dear, let us begin with a embryo sac. This embryo sac is also called it as a female gametophyte. Okay. Now, dear, let me show you first the diagram of this. See, this all are the processes which we are going to study under a megasporogenesis. Megasporogenesis is also called as a female gametophyte, and it is also called as an embryo sac. All right. Now, this megasporogenesis, you know, the megaspore means the female spores are formed by a megaspore mother cell, which is major mother cell. And it is divided by whom? It is divided by a meiotically, not mitotically. It is divided meiotically. And hence, this process is called as in megasporogenesis. So, this whole of the cycle we are going to study now soon. All right. And before that, if I'll show you, this is a diagram of the embryo sac, which is it is going to lead finally at a what? Finally at a development of the a proper formation of embryo sac, which we also called it as in female gametophyte. Now, in this embryo sac, dear, when it was young, we called it as a nucellus, and this embryo sac, when it gets mature, why? How come it get mature? How come we get its signal? It consists of an chalazal end, and it consists of a micropylar end, and it is consisting at a downward direction three cells. Three cells are whom? The two cells are synergids, and in middle there is a present of an egg cell. In an upward direction, if you will see into a embryo sac, we called it as an antipodal cell. And into a center we called it as in polar nuclei, and hence it is also called it as in central cell because it is consisting of an polar nuclei. It is showing the direction to both of this upward and downward cell, and therefore they are present into a center cell, so it is called as in central cell also. Understood, Siddhant, earlier?
Yes, ma'am.
Yes. Now this is a proper diagram of our embryo sac. Now let us begin. Now, this nucellus, when it gets developed, we called it as an embryo sac, which we also called it as a female gametophyte. It is normally this ovule, the pollen tube, enters from the micropylar end, which is present in towards a downward direction. Therefore, this embryo sac, the shape of this embryo sac is ovule, and therefore, oval, and it is haploid in condition, and it consists of in how many cells? It consists of in 7 cells and 8 nuclei. The embryo sac consists of in 7 cells and 8 nuclei. So we called it as in what? 7-celled and 8 nucleated structure. Why we called it as in all these things, that is 7-celled and 8 nucleated structure? Let us begin. This embryo sac, which we called it as an female gametophyte, it consists of an 3 majorly cells. Firstly, we called it as an egg apparatus. Secondly, we called it as an secondary nucleus. And thirdly, we called it as an antipodal cells. Let us begin with the first cell, which we called it as an egg apparatus. This egg apparatus, what? It is present towards where? It is present towards a micropylar end. And therefore, this micropylar end, which it consists of an egg apparatus, it consists of 3 cells in the middle towards the downward direction. In middle, if you will see, it is a lateral middle, it is a presence of an synergid, and this synergid is surrounded by a 2 cell, which we called it as an egg cell. Therefore, what is the function of this egg apparatus? The function of this egg apparatus is, during fertilization, this egg cell fuses with one male gamete. How many male gametes are present, Siddhant? 2, right? Now, this 2 cells, so first of this male gamete have been got fused with the help of an egg cell, right? Now, this egg cell is going to fuse with one male gamete. It will form a diploid zygote. Therefore, what is the role of this synergid? The role of the synergid is it is providing a nourishment and it is giving a support during the fertilization to the egg cell. That's why, firstly, I shown you the diagram so that the view should be clear towards you. Now, this is a diagram of an embryo sac. If you will see, this is in middle, there is a present of an egg cell. So there is a presence of in 2 male gametes. Hence we have saw in a development of male gamete. तो एक मेल गैमेट है वो इस egg cell के साथ फ्यूज होगा और डिप्लॉयड सेल बनाएगा, राइट? और बाकी के ये जो 2 laterally surrounded, किसको सराउंड करके है? एग सेल को. तो ये जो दो सिनर्जिड्स है वो क्या करेंगे? वो सपोर्टिंग मीडियम प्रोवाइड करेंगे, नरिशमेंट प्रोवाइड करेंगे फर्टिलाइजेशन के वक्त. किसको? ये एग सेल. समझ में आया डियर? एग सेल का क्या रोल है एम्ब्रियो सैक में? ओके सिद्धांत?
To fertilize the male gamete.
Yes, to fertilize the male gamete. Understood? So 2 male gamete, 1 egg cell. So in embryo sac, present downwards the 3 cells, that is the 1 egg cell has got fused, okay, with 1 male gamete. दो बाकी के synergids वो supporting medium में रहेंगे और उसे nourishment provide करेंगे fertilization के दौरान. All right. Now let us move further. The secondary nucleus. Secondary nucleus, it is present at the center of the embryo sac, जिसे हम कहते हैं polar nuclei. Okay, so this polar nuclei, it is nothing but what? It is a secondary nucleus. Done with the writing, Siddhant?
No, ma'am.
Okay, write it down here.
Done, ma'am.
Done, dear? Okay. Now let us begin with the secondary nucleus. This secondary nucleus is present in the center of the embryo sac. All right. And the polar nuclei which it are present, right, they both get fused. Okay. It is formed by fusion of two polar nuclei. So whenever these two polar nuclei get fused together, we called it as in secondary nucleus itself. Therefore, these polar nuclei, which are a secondary nucleus, they are a diploid cell. And what is the function? During the fertilization, the second male gamete is present. It fuses with the secondary nucleus, which is a polar nuclei. And what it form? It form a triploid primary endosperm nucleus, which we called it as in TPEN. अंडरस्टूड सिद्धांत दो मेल
Yes,
गैमेट्स
ma'am.
थे यस पहला मेल गैमेट एग एपैरेटस के साथ मतलब बीच के एग सेल के साथ फ्यूज हुआ तो बनाया उसने डिप्लॉइड जाइगोट दूसरा मेल गैमेट और बाकी था तो वो जाके सेकेंडरी न्यूक्लियस मतलब हमारे पोलर न्यूक्लियाई के पास जाके फ्यूज हुआ तो हम उसने बनाया ट्रिप्लॉइड प्राइमरी एंडोस्पर्म न्यूक्लियस जिसे हम कहते हैं TPEN, right? So in this way, what it is, in this way the proper formation of and the proper utilization of the male gamete has been done, and this both the male gametes and the both the diploid cell and TPEN plays a major role during the double fertilization activity. Therefore, the third type of a major cell, we have a antipodal cell. The antipodal cell you can see over here, which is present near the chalazal pole, that is near the chalazal end. Okay. Therefore, now this antipodal cell, it consists of in three cells, which is present near the chalazal end. What is the function? This function of this antipodal cell is therefore what it is. It shows a 80 embryo sac in monosporic and in endosporic. Now we everybody know that the male gametophyte and female gametophyte both are endosporic because it is present inside the ovule, right? क्या है जो मेल गैमेटोफाइट है वो इनसाइड द पोलन ग्रेन प्रेजेंट है, जो फीमेल गैमेटोफाइट है वो हमारे इनसाइड दी ओवरी मतलब ओव्यूल्स के यहाँ पे प्रेजेंट है, राइट? अगर हम ऐसे देखने जाए कुल मिलाकर हर एक प्लांट में कुल मिलाकर हमें 80 एम्ब्रियो सैक मिलेंगे एंजियोस्पर्म में, राइट? जो कि एक एक्सेसरी सेल्स के नाम पे वो फंक्शन करते हैं. And therefore this all these 80 cells individually they form how many? 7-celled and 8 nucleated set of the cells. And when the plant is forming the 7-celled and 8 nucleated set, we called it as an polygonum type of ovule. Understood, Siddhant? समझ में आया यहाँ तक? लिख लो, फिर मैं आपको मेगास्पोरोजेनेसिस का डायग्राम एक्सप्लेन करती हूँ. ओके, राइट इट डाउन.
मैम
या
आई हैव अ डाउट मैम,
या
तो हर बार प्लांट में डबल फर्टिलाइजेशन नहीं होगा?
हर बार प्लांट्स में डबल फर्टिलाइजेशन होता है बेटा. Okay, it is a process. See, we have to keep the heredity of the plants going on, isn't it? Though the plant, it is a tree, whenever the plant sapling is small, from the plant sapling is small till the lifespan, whole of this activity is going to happen again and again, double fertilization. Because is it like that, Siddhant, only one fruit we get it at a time on the tree? No, we get it multiple times fruit, right? In each and every seasonal time, isn't it? So this double fertilization activity has been carried out all the times. Like for example, this mango season, right? In summer season, we get it as in mangoes, right? So when there is a summer season, this is a proper time in India to get a mango. So definitely at that time, the double fertilization is carried out every year into that single plant. Understood? That time we get it as in food. अगर ऐसा होता तो क्या होता, हमें सिर्फ एक ही बार मिलता, उस मैंगो प्लांट की भी लाइफ स्पैन कम होती. बराबर है कि नहीं? लेकिन ऐसा नहीं होता. जो फ्लावरिंग प्लांट्स है, जो फ्रूट बेयरिंग प्लांट्स है, उनमें हर बार जब फ्लावर बनना है तो डबल फर्टिलाइजेशन होगा. अगर वो फ्लावर से फ्रूट में कन्वर्ट होने वाला प्लांट है तो उसमें होगा आपका डबल फर्टिलाइजेशन एक्टिविटी. इट इज कैरिड ऑफनली बच्चा. ओके, नॉट ओनली सिंगल टाइम. अंडरस्टूड सिद्धांत? या.
Yes ma'am.
Done with the writing?
Yes ma'am.
Yeah. Okay. Now, next. This is a diagram for a megasporogenesis, dear. Now, the first thing is what megasporogenesis, which has been shown in a diagrammatic representation. Over here, you can see that is what there is an anatropous ovule. It is consisting of an ovule with a diploid megaspore mother cell. अब हमें मेगास्पोरोजिस का ऑलरेडी डेफिनेशन पता है कि ये जो मेगास्पोर मदर सेल है, it is going to make a megaspore that is female megaspore. Okay, therefore अब ये जब मेगास्पोर जो है बना है, therefore मेगास्पोरोजेनेसिस का डेफिनेशन क्या कहता है सिद्धांत? It is going to divide mitotically. Okay, yes or no सिद्धांत? बराबर. मियोसिस मियोटिकली होगा, सॉरी आई सेड मियोसिस मियोटिकली होगा, बराबर है कि नहीं? है ना? तो इसीलिए क्या होगा, यहाँ पे ये जो मेगास्पोर मदर सेल है, ये क्या होगा, ये मियोटिकली डिवाइड होना स्टार्ट होगा. When it will form with a meiotically, what will happen in a second megasporogenesis? How many megaspores will be formed? It will be formed tetrad. Means the 4 megaspore formation will be happening, right? And therefore they all will be in a haploid condition, because over here, which is our megaspore mother cell, it has been what, it is into a diploid condition. ये मियोसिस थ्रू डिवाइड होकर हमें यहाँ पे 4 सेल्स देंगे मेगास्पोर के, 4 फीमेल गैमेट्स मिलेंगे, लेकिन वो सारे रहेंगे हेप्लॉइड कंडीशन में. Later on, when the further development, when it will be starting place, what will happen? The megaspore means the whole of this ovule has been formed with what? It has been formed with a chalazal pole. It has been formed with a micropylar region. And therefore, for a further megasporogenesis, for a further megaspore in a functional condition, rest all megaspores are going to get what? Degenerated. One of the megaspore is going to move further, and rest all which are what? Rest all which was present, they are going to get degenerate slowly. Now the same one megaspore, we have took it over here in a ovulated condition. I called it as a functional megaspore because it is going to carry out whole of the activity for my female gametophyte to carry on, right? तो ये जो एक मेगास्पोर फंक्शनल में है, उसके पास क्या है, छोटे छोटे सेल्स है जो कि अभी भी नॉन फंक्शनल में है, क्योंकि ये क्या है, यंग मेगास्पोर है, और साथ में उसके पास न्यूक्लियस प्रेजेंट है. अब ये जो मेरा फंक्शनल मेगास्पोर है, ये सबसे पहले न्यूक्लियर डिवीजन में जाएगा. Okay, so firstly it has been carried out a first nuclear division, and this first nuclear division, it has been carried out, you got a 2 nucleated embryo sac. How you got this 2 nucleated embryo sac, Siddhant? Because this single megaspore, for getting it 2, it has went into a mitotic division. Okay, mitotic cell division में ये गया, इसलिए पहले न्यूक्लियर डिवीजन में हमें एम्ब्रियो सैक में 2 सेल्स मिले, मतलब 2 न्यूक्लिएटेड स्ट्रक्चर्स मिले हमें. ओके, अब यही 2 न्यूक्लियसिस में क्या होगा, मियोसिस कैरी होगा, जिसे हम कहेंगे सेकेंड न्यूक्लियर डिवीजन. तो ये जो सेकेंड न्यूक्लियर डिवीजन है इस ओव्यूल में जो हुआ है, एनाट्रोपस ओव्यूल में हुआ है, उसमें क्या होगा, उसमें 4 न्यूक्लिएटेड मींस एक एम्ब्रियो सैक में 4 न्यूक्लियस बने। कैसे? मियोसिस के मदद से। In a third nuclear division, hence we want a 8 nucleated structure, right? So therefore ये भी सारे सेल्स है जो इंडिविजुअली है, ये जो दो दो सेल्स है जो कि डिप्लॉयड कंडीशंस में है, they will be going under a meiosis and they will form a 8 nucleated embryo sac. Now in this 8 nucleated embryo sac, यहाँ से from the upper side, from a chalazal end, one of the cell is going to transfer in a center, लोवर माइक्रोपाइलर एंड से, जहां पे हमारे 3 सेल फॉर्मेशन है, अभी के लिए 4 है, यहां से 4, ऊपर से 4, नीचे से 4, चलाजल एंड से एक सेल मिडिल में ट्रांसफर, फ्रॉम हियर फ्रॉम अ माइक्रोपाइलर एंड यहां पे मिडिल में ट्रांसफर, therefore both of these cells are present over here, इसलिए मैं उसे क्या कहूंगी, shifting of nuclei. So from upper where there was an 4 from chalazal end, one is traveled in between, lower at a micropilar end, one has been traveled in between, we called it as in polar nuclei. And this polar nuclei further it is going to get fused and therefore it is going to become a secondary nucleus, जो कि हमारा second male gamete है, उस secondary nucleus के साथ जाके fused होता है और बनाता है triploid primary endosperm nucleus. Okay dear. लेकिन सॉरी, यहाँ पे शिफ्टिंग ऑफ द न्यूक्लियस कैरी आउट है, अभी के लिए हमें सिर्फ पोलर न्यूक्लियाई मिले हैं, बराबर. Now this whole my megaspore, I called it as a female gametophyte, it is under a functional stage. All these cells have been organized into a proper manner. So I got at a 3 stage, that is at a chalazal pole, at a upper stage I got a antipodal cells which all are in a haploid condition. Middle I have my polar nuclei, which is my secondary nucleus, I can call it as in, and at a lower I have a 3 cells which I called it as in what, I called it as in micropilar end. In middle this 3 cell, it is a egg cell which is under, it is going to again play an important role with a whom, with a male gamete, and these 2 cells are what, these 2 cells are synergid cells जो कि इस egg cell का supporting medium है और क्या है, इवन सपोर्टिंग मीडियम के साथ वो उसे नरिशमेंट प्रोवाइड करेगा. तो एक्सेल एंड सिनर्जिड्स हम इन दोनों को मिलाकर बुलाते हैं एग एपैरेटस. अंडरस्टूड? Therefore,
Yes.
this it is called as in what, Siddhant? It is called as in 7-celled and 8 nucleated embryo sac. All right. 1, 2, 3, 4, 5, 6, 7, 8 पोलर न्यूक्लियाई को पकड़ के 7, 8 कितने न्यूक्लिएटेड एम्ब्रियो सैक है? 8, राइट. उसमें से अगर आप सेल्स काउंट करो तो कैसे करोगे, मुझे बताओगे?
3 antipodals,
हाँ
1 polar nuclei, 2
हाँ
synergids, and 1
हाँ.
egg apparatus.
Okay, so what this is, 3 antipodal, 3 of this lower cells, 3 plus 3, 6.
6.
आगे जाके दोनों भी पोलर न्यूक्लियर फ्यूज्ड हो जाएंगे, एक बनेगा सेकेंडरी न्यूक्लियस, 3 plus 3, 6, सेकेंडरी न्यूक्लियस 6 plus 1, 7, 7 celled and 8 nucleated embryo sac. Understood?
Yes, ma'am.
समझ में आया? This whole embryo sac we also called it as a megaspore. This is our mother megaspore. Okay, mother celled megaspore, and we also called it as a female gametophyte. Understood, Siddhant?
Yes, ma'am.
अंडरस्टूड? यहाँ तक समझ में आया आपको कैसे बना? हाँ, अब यही हम पूरा एम्ब्रियो सैक अभी पढ़े. अब I'll tell you how to write it down in exam. In exam, if it is asked, okay, describe the structure of anatropous ovule. All right. Firstly, what are you going to do? You're going to make this diagram with the proper labelings. All right. उसके बाद आपको आंसर स्टार्ट करना है गाइनोशियम से, लेकिन आपको लिखना कहाँ तक है एनोट्रोपस ओव्यूल में डियर? यू हैव टू जस्ट राइट इट डाउन टिल दी फीमेल गैमेटोफाइट. ये जो मेरा पॉइंट है ना, यहाँ तक न्यूसैलिस तक, ये न्यूसैलिस के पूरे मेजर पॉइंट्स आपको 4th पॉइंट तक, मतलब फीमेल गैमेटोफाइट तक ही लिखना है. कभी जब आपको डिस्क्राइब दी एनोट्रोपस ओव्यूल पूछा जाएगा तभी. अंडरस्टूड सिद्धांत? लेकिन अगर आपको पूछा जाएगा डिस्क्राइब फीमेल गैमेटोफाइट, तो सबसे पहले आप ये डायग्राम बनाएंगे मेगास्पोरोजेनेसिस का। आपका आंसर स्टार्ट होगा स्टार्टिंग से, जो कि क्या है, हमारे इसमें गाइनोशियम से आपको आंसर स्टार्ट करना है, एंड यू हैव टू एंड योर आंसर नियर दी पॉलीगोनम टाइप। समझ में आया सिद्धांत? अंडरस्टूड? एनाट्रोपस ओव्यूल बोला तो एनाट्रोपस ओव्यूल का डायग्राम बनाकर सिर्फ फीमेल गैमेटोफाइट तक एम्ब्रियो सैक कैसे बना वहां तक लिखना है आपको। लेकिन अगर फीमेल गैमेटोफाइट पूछा तो आपको पूरा आंसर लिखना है विद फीमेल गैमेटोफाइट, लेकिन डायग्राम आप ये बनाएंगे। ओके,
Yes, ma'am.
all right, you can make it in a cyclic manner also, however feasible to you, right? Or you can make this diagram also, it's your wish, but this is a criteria and proper thing to write down the answer. Okay dear, then and then you get it full 5 marks. Okay, understood Siddhant? All right, now write it down, homework dear. Only 2 questions I'll give you in homework.
Yes, ma'am.
Shall I say? Okay. Describe the male gametophyte. Describe the male gametophyte with proper diagrammatic representation. With proper diagrammatic representation. Second question. Comment on megasporogenesis. Comment on megasporogenesis. ओके, जहाँ पे मैंने आपको कमेंट बोला है आपको पूरा आंसर लिखना है मेगास्पोरोजेनेसिस में। ओके, एनाट्रोपस ओव्यूल भी, और आपको ये एम्ब्रियो सैक के बारे में भी डायग्राम आपको यही डिस्क्राइब करना है। ओके, ऑलराइट, whichever feasible for you. If you have a cyclic representation, you can draw that also. If you found this easy, you can draw this also. It's upon you. Okay, but the technique of the answer should be the way I said you. All right, Siddhant. So today we have completed with a major thing, that is a male gametophyte. We have completed with the anatropous ovule and megasporogenesis. All right. Now in the next lecture, most probably we will be completing with a double fertilization, pollen pistil interaction, dear, right? And the small part of this, that is the embryo sac, small part of this I'll be giving you. That is what is an archisporium, what is a megaspore mother cell. Megasporogenesis already you know. This is easier. Only I want to say that what is a mega archisporium. All right, Siddhant. Then I'll be completed with double fertilization, pollen pistil organization, and how many type. I'll be telling you about the endosperm and the types of endosperm. All right, and most probably I'll be trying to complete about the pollination activity also. Okay, Siddhant, it will take little bit longer time because the reason behind that, you are keeping the notes also, right? I'll be giving you the notes. I'll be forwarding you the notes. I have got your mail ID dear yesterday itself. Okay, and from next time onwards, I have got your number from Hemant sir. Okay, Siddhant, so you'll be sending me the homework itself or whatever query you have itself on my number. Okay, WhatsApp me. All right,
Ma'am,
dear.
in the male
Yeah.
gametified answer,
Yeah.
I have to write from development of male gametocyte
Yes.
to after pollination of the stigma.
Yes, yes, after pollination of stigma. See, I'll tell you. Wait, I'm going towards that slide only. Okay, dear, wait, I'll tell you. See, you have to draw this diagram first of male gametophyte. All right. After drawing this diagram, you're going to begin with your answer. That is, you're going to write all those things. Okay. You have to write down till the after pollination of the stigma. Means you will be completing till 2 male gametes and cytoplasm represented male gametophyte. ये पूरा आंसर है मेल गैमेटोफाइट का. ओके सिद्धांत, ऑल राइट. Any kind of inquiry, dear, in today's lecture? Any kind of
No, ma'am.
inquiry? Okay. All right. You have understood today's lecture? You have understood today's lecture? What? No? Yes or no,
Yes, ma'am.
Siddhant? Yes. So you nodded your head in
Yes,
this
yes.
way. Okay. Okay. Fine, Siddhant. All right. Thank you for attending the lecture, dear. Thank you for your cooperation. Have a nice day ahead. If you have any kind of inquiries, you can WhatsApp me. All right, dear.
Bye, ma'am. Thank you, ma'am.
Bye, beta. Yeah. Thank you, dear. Thank you.
Ma'am,
Yeah.
when
Please.
do I have to submit this homework tomorrow?
Yeah. Till tomorrow evening, dear.
Okay,
All
ma'am.
right. Tomorrow we don't have lecture, right? So till tomorrow evening, you can submit it to me. You can directly forward it to me. If it
Okay,
is
ma'am.
finishing prior before, you can send it to me. Not an issue. Okay. Tomorrow till evening before 6 o'clock, you send it to me the homework. All right, dear. Okay. Ciao.